why the fish in rivers jumps like that? Well, in the 1970s, two types of carps called the silver carp and big head carp were imported from China to the US. They were originally brought in for farming and sewage treatment. But unfortunately, a flood happened in the 90s, and the carps escaped into the rivers and found themselves a new habitat. But was it originally theirs? Let's find out. Silver carp and big head carps are commonly called Asian carps, but they're actually different fish with very different behaviors. The most notable difference is that only the silver carps jumps out of water when they're startled. Both these type of fish reproduce rather quickly, and therefore they're overly abundant in many Midwestern US rivers. But without many predators and with a high rate of reproduction, there will soon be too many carps. They will compete with native species for food and habitat, altering the natural food web of the river. The problem with these non-native fish moving northward within the Mississippi is not only interfering with the river's ecology, but eventually invading the 4 Great Lakes if they enter from Lake Michigan. Luckily, the key to preventing Asian carps from entering Lake Michigan lies in the Chicago area waterway system. Electric barriers and physical structures could possibly prevent fish from moving through the system. But how do we know if efforts to prevent the movement of Asian carp are working? When silver carp are concentrated in a small river area, they're easy to see. But scientists might not be able to detect 1 or 2 silver carp that slip past barrier, right? How will researchers obtain evidence to determine whether these barriers are preventing the Asian carp from invading the Great Lakes? Simply, biologists will use DNA analysis to search for evidence of the Asian carps. Each species has a unique DNA fingerprint. Scientists could analyze the DNA in a river area to look for a unique combination of DNA associated with the particular species. In this case, biologists will compare the DNA in the river water to the known DNA for each species. Scientists can then assess invasion, but in order to collect DNA to analyze, researchers must filter the organic matter out of each water sample before they can start detecting the carp's DNA. Each water sample does not contain a sufficient amount of eDNA. Therefore, they amplify the amount of DNA with a process called PCR, which then allows them to use gel electrophoresis to analyze the DNA content for the presence of each species. This process also yields an ample amount of DNA to use in further analysis.
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
Now, after the DNA is amplified using PCR, samples can be analyzed using gel electrophoresis. During the process of gel electrophoresis, researchers use restriction enzymes to cut DNA into fragments or bands of varying size, each of which bears a particular genetic code. Each restriction enzyme recognizes a specific base pair sequence on both strands and cleaves the DNA at that point. Here's another video to further elaborate this gel electrophoresis.
Once DNA has been extracted from a sample and a section bearing useful code such as a VNTR or STR is isolated, the next step in DNA fingerprinting is to measure each strand and count the number of repeated sections. To do this, scientists use a technique called gel electrophoresis, which uses an electric current to push strands of DNA through a slab of gel-like material. Because each bit of DNA is negatively charged and subject to an equal electric force propelling it to the positively charged side of the gel, smaller pieces move faster than larger ones. When the current is removed, the gel is photographed to reveal how far each bit has migrated. By comparing the bands produced by the DNA samples of interest with bands produced by standard samples whose sizes are already known, the precise length of each DNA fragment can be gauged.
In other words, the restriction enzymes cut each unique DNA sample at different locations. They produce different sized bands of DNA. The pattern of bands produced is different for each species, because each species' DNA has a unique sequence of base pairs. Therefore, the size of the bands produced during gel electrophoresis allows scientists to see whether DNA from silver carp, pighead carp, or both is present in a sample. While many types of sampling methods are available to researchers, eDNA provides an advantage over some of the more traditional methods. So why is it possible to use a DNA marker to detect the presence of silver carp in the environment, for example? Simply because certain sequences of DNA are found only in silver carp. So, could the video of the jumping carp from earlier be a video from Lake Michigan in the near future? Can biotechnology protect the Great Lakes from these flying fish? Well, scientists and engineers are working hard to prevent this from happening. Biotechnology, including eDNA surveillance, can provide scientists with data to help them identify areas that might have been invaded by non-native Asian carp. Fishery managers can then focus on protecting native fish in these areas. Thus, that's the end of the lesson.
Thank you so much for watching. See you next time.